So it's my pleasure to introduce Michel Sadelain, our final speaker. He's the director of the Center for Cell Engineering at Memorial Sloan Kettering. He is one of the pioneers of the CAR-T field, which has absolutely revolutionized the cancer immunotherapy area in the recent years. He's the founder of Therapeutics, and the title of his talk is Using iPSC-derived T cells for cancer immunotherapy. So please give a warm welcome to Michel Sadelain.
Thank you. Thank you. Well, thank you for the invitation. I think we all agree that a key to develop efficacious but also safe drugs for cancer is through specificity. But rather than designing chemicals or small molecules, some of us have, of course, focused on something nature offers to us, which is an adaptive immune response that's exquisitely specific. Furthermore, T cells are not just specific, but they can persist for a long time through in the form of memory, and in particular fight cancer over enough time. However, while they are specific and long-lasting, they're not always potent enough, and this provided a rationale many years ago for wanting to engineer T cells. And in fact, for me, it was over 25 years ago when I was a postdoc, one floor below Rudolf Jenisch, that I started trying to engineer T cells and found that you could do that with retroviral vectors. The rationale really is that there are many things that you could accomplish by engineering T cells that deal with their specificity, their functional properties. And I'll just run you through a quick list here. One is to overcome central immune tolerance. We have a rich repertoire generated through VDJ recombination, but it's not infinite, and it can undergo attrition in patients with cancer. Well, in a single step, of course, you can generate T cells of any specificity of your choice if you have the right receptors. Using CARs, which you've already heard about, you can circumvent HLA downregulation, which is a major mechanism of tumor escape from natural T cell responses, where the T cell receptor is used to recognize HLA peptide complexes on the surface of tumor cells. With T cell engineering, you can target both CD4 and CD8 T cells. CD4 helper cells don't always interact with tumor cells because they don't often express MHC class II genes. But again, an engineered T cell can provide help at the doorstep at the tumor site itself. You can broaden T cell reactivity beyond the HLA peptide complex when you use CARs and, for example, target carbohydrates and glycolipids. Ideally, you'd want to target cancer stem cells or tumor-initiating cells, and of course that's the key to a curative therapy. Now, augmenting T cell potency, I've already mentioned, and I'll talk about for the next few slides before getting to iPS cells, and that's a major effort in the field. And controlling T cell longevity is also critical. It should be long enough to allow for the eradication, the ideal eradication of a tumor, but sometimes T cells have to be abrogated because of their side effects. And lastly, and this is key for today really, thanks to genetic engineering and iPS technologies, I think we're reaching the time where we can explore alternative T cell sources, and by that I mean non-autologous cell sources. So you've already heard about CARs from Dan, and I just want to remind you that the T cell receptor is the physiological receptor. This alpha-beta heterodimer recognizes HLA peptide complexes. Itself, it does not signal. It is associated with the CD3 complex, which comprises 4 independent gene products, epsilon, delta, gamma, and this green here zeta chain. The TCR itself is critical to initiate a response, but not sufficient to direct a productive immune response. And it is assisted by a myriad of costimulatory receptors that fall in 2 main families, members of the immunoglobulin superfamily, such as CD28, or members of the TNF receptor family, such as 4-1BB. I'll skip the history of CARs, but what we described several years ago now is what we then called, when we introduced the CAR nomenclature, a second-generation CAR. And it has 3 canonical components: an scFv here that recognizes, again, cell surface antigens, An activating domain, most commonly borrowed from this CD3 zeta chain, and this third very essential component, the costimulatory signaling domain, that increases persistence and function of these T cells. So to test these in the lab, and wanting, of course, to bring this to the clinic, we had to identify an appropriate target. And over 15 years ago, we set our eyes on CD19. It's a cell surface molecule. It's expressed on the majority of lymphomas and leukemias. We speculated at the time that it might be expressed on the tumor-initiating cell for those cancers. We knew it was expressed on normal cells, but thought that the B-cell aplasia that could be caused by CAR-T cells would be tolerable because rituximab, for example, induces B-cell aplasia, and physicians are accustomed to managing this side effect. And lastly, and perhaps naively, we thought that by depleting B cells in these patients, we might curtail any eventual immune response that patients could develop against the CAR, because it is a chimeric molecule, and the first ones actually use scFvs derived from monoclonal antibodies. So anyway, this is the first demonstration in the literature that a human T cell could be engineered with a CAR, expressing a specific, in this case for CD19. And here's a Raji tumor, a CD19 purulent lymphoma, an FDG PET scan showing you the tumor in the marrow and calvarium. And you see that a single infusion of these T cells could induce long-term remissions and cures. And then we found that this was also the case in patients with acute lymphoblastic leukemia. The studies at Memorial focused on adults, and this is from our first report in 5 patients. I'll just highlight the two more dramatic of these 5 patients. All of them responded to therapy. Here's the FACS analysis of bone marrow on day -4 for subject 4. All this here is CD19 positive or CD10 positive as well. It's the blasts that you see above. And you can see that on days 11, 24, 59 after T-cell infusion here, the complete obliteration of any CD19 positive cell in the marrow. Likewise, in subject number 5 here, you see again 80% blasts in this patient that you see up above. And on days 22, 29, 50, the complete obliteration of any CD19 positive cell in the marrow compartment, as well as blood and elsewhere. So relapsed chemorefractory adult ALL is lethal. These patients have weeks or months to live. A bone marrow transplant is not attempted at this stage because it is known to be inefficient. And here we are with these complete responses induced by a single infusion of T cells. And they're not just a complete response, they're really molecular responses. And in these 2 individuals, again, I'll highlight here the deep sequencing data. You see at day -4, 2 million transcripts here of this malignant clone per fixed amount of genomic DNA, going down by day 11, 24, and somewhere between day 24 and day 59 here, you see this becomes negative. It's even more dramatic in subject 5, where you see here 3 million transcripts at day -1, and by day 8 already, the tumor is undetectable by deep sequencing. Of course, we were pleased that the editors of Science, when they celebrated cancer immunotherapy to be the breakthrough of the year back in 2013, not only highlighted, of course, checkpoint blockade, but also this emerging technology based on CD19 CARs. Just a quick update with today, 45 patients. We're actually closer to 55 today, but recently updated at ASH, we're 45 adults treated on this protocol. The overall CR rate is 82%, and of those, 83% go into this MRD negative state. And I'll show you the importance of that in the next slide. Of interest to us is the time that it takes to reach the CR, which is on average 3 weeks, sometimes as fast as 8 days, as in the example I just showed you, and sometimes taking 6 weeks. And that tells us that whatever cells we make should persist for at least 6 weeks in patients with ALL. And here's, if you look at the survival data, these data are still maturing, but if you look at those patients that go into CR, clearly you see that slightly more than the majority of these patients will likely survive for at least a long time, whereas those that have residual disease, those 10 to 15% that I showed you that have detectable tumor, will relapse and will die from their disease. You might ask, does this work better in some individuals than others? Well, if you look at disease burden, whether they had previously a transplant or not, the number of prior chemotherapies that they might have received, 2, 3, 4, or more, at their genetic status, Philadelphia chromosome or p53 mutations, or if you look at their age, young adults or older adults, there's no difference, and this points to how robust it is to take one's T cells, engineer a CAR, and then reinfuse them. Another element that underscores the robustness of this approach is that after we published our mouse models, two other groups were interested in performing similar trials at Penn and at the NCI, and more groups since then. And here are the CARs that they used. There's the original CD28, CD3 zeta-based CAR from our group, and the 41BB CAR that had been described by Dario Campana's group at St. Jude. That's the construct that was used at Penn, and the Hutch now has their own 41BB CAR, and the NCI and Baylor later designed their own CD28-based CARs. The only point I want to make here really is that all of these groups reported very similar outcomes in ALL, whether in adults or in children, again pointing to the robustness of this approach. And so as everyone here knows, this has sparked enormous interest in cell-based therapies that until then had been always sort of an academic expertise, and many more medical centers as well as companies now have formed to further develop this technology. And before going to iPS cells, I just want to make highlight a study where we are, of course, aiming to make yet better CAR-T cells, not just to make them better and to tackle solid tumors, but the point I want to make too is that, which I'll make right away, is that with better CAR-T cells you'll need far fewer, which I think will also address this issue of cell doses and manufacturing. The cells that I show you we're going to bring to the clinic in August, and we think that you may need 10 times fewer cells to infuse. So again, this is this first, second, and third generation CAR. And here's what we are developing intensely at Sloan Kettering. It's really using the CAR-T cell to not just kill tumor cells, but affect the tumor microenvironment. And so we have CAR-T cells. We have an ovarian trial that we just opened where the CAR-T cell secretes cytokines. We have this trial that I'll opening soon where the CAR is co-expressed with a costimulatory ligand, which I'll show you in a minute. And we're also developing CAR T cells that secrete various scFvs, again, selected to target checkpoints in the tumor microenvironment. So I just want to show you how we test and improve these CARs, and I'll quickly run you through 7 different CARs here. They're all specific for CD19. This is the so-called first-generation CAR with the zeta chain signaling domain. These are the more famous second generation, and these are the 2 that are in the clinic today. The red one is CD28, and the green one is 41BB based. And below here are 4 different CARs that aim to recruit all 3 pathways in parallel. I mean by that the zeta chain to activate and both the 28 and the 41BB. This is a third-generation CAR here on the left. And here you see this CD28 CAR, again here co-expressed with the ligand for the other receptor, which is 41BB. Here is a 41BB CAR co-expressed with a ligand for CD28. And to the right here is that first generation CAR shown here, co-expressed with both ligands. And I'll just show you 2 slides really, comparing the potency of these T cells in animal models. And so this is an ELM6. It's a human acute lymphoblastic leukemia. It's established in these mice, and then they're infused with 400,000 CAR T cells. The first generation CARs I showed here, these is 120 days on the x-axis. First generation CARs ineffective, as I said before. Both second generation CARs here induce tumor delays. Again, if you use 1 million cells, 5 or 10 million cells, as you'll often see in papers, they're both therapeutic and curative. Here they induce these delays, and of course a lower dose is not helpful, and you see the outcome here, 200,000 CAR T cells per mouse. So here are these new CAR designs. The triple fusion is actually very disappointing. It's a great killer, it makes lots of cytokines, but expands poorly. And this design, which I won't show you here, expands massively. You almost think these mice have leukemia, but the cells are not so effective. And of these 7 combinations, these 2 emerge as the winner, and this is really the 1928Z currently in our trials, co-expressed with 41BB ligand. There's tumor progression before regression because we give a low T cell dose, and that's the essence of the stress test. And we can now treat these mice, in fact, with 50,000 CAR cells per mouse. So this is what we're bringing soon to the clinic, and we think this will greatly reduce. We don't know yet, of course. We have to go to the clinic and find out. But based on these animal studies, we think that just the T-cell dose will enormously decrease. So another major interest of our program is to explore really the potential to not just make CAR T-cells from autologous donors, but to actually make the T-cell in vitro. And so we've established this system similar to what Dan Kaufman just showed you. We actually started from iPS cells derived from T-cells, such that we would know what T-cell receptor they have owing to prior VDJ recombination. And the CAR is expressed from lentiviral vectors in these cells, co-expressed with DS-RED, which you see below here, with continued expression through these different phases of differentiation. And it's a fairly classic culture with a first step of mesoderm induction, then hematopoietic specification. And then these cells, around day 10, go on the OP9-DL1 cell line that was previously described by Juan Sinica-Fluker to induce T cells in mouse and also in human. So the cells that we get after these cultures, around 25 days, you can overlay them. They have a CD19 specific CAR. You can see here if they go on cells 3T3 fibroblasts expressing CD19, they form these nice clusters. To the lower right here, you see that makes them induce CD69, CD25. They also secrete cytokines, TNF-alpha, IL-2, interferon gamma. We can expose them to these artificial antigen-presenting cells weekly here, and you see these cells proliferating. So they are antigen-specific, and they appear to behave like lymphocytes. In an in vivo model, using the Raji-Burkitt lymphoma intraperitoneally and infusing 200,000 of these T cells, this is what we found. This is 3 mice of larger cohort, representative of all of them. Here's the initial tumor, IP. You can see it disappearing, although it does come back in this mouse. And this is very similar to what you get when giving 200,000 T cells from the same donor from which the iPS line had been established. So they're isogenic, if you like, in a gross way in this regards. And you might ask, why are we comparing them to gamma delta cells and not just the more common alpha beta cell type? I'll explain that in the next slide. Well, the alpha beta cells, of course, are the best lymphocytes in this setting, and you can see all these mice are cured. And the gamma delta T cells are really quite good, although not quite as good. You see a relapse here, very similar to the iPS-derived T cell. So why did we perform this comparison to gamma delta T cells? Well, upon close examination of these cells, we found that, yes, they have lymphoid signatures, but they are shown here, the 1920 T-IPSC T cell. You can see here these red boxes here that are akin to the gamma delta T cells and other in its cell types. So these cells are T cells, but they have many features of a gamma delta T cell. They're clearly not NK cells, which you see here is very different from what we see. But they have these innate features, although they do express an alpha beta T cell receptor on their surface, but internally they are wired like a gamma delta T cell. And so why is that? Well, there are several cues to point out that this differentiation wasn't quite like the other ones. If you use cord blood CD34 positive cells and perform this in vitro differentiation, it takes a certain time, often 35 days to 40 days, sometimes a little more, to get T-cell, TCR alpha beta positive T cells in these cultures. Very similar culture using OP9-DL1. With the TIPS-derived cell, the T cells come much more quickly. As early as day 15, you already see all these alpha beta receptor positive cells. Now, when you do the differentiation from cord blood cells, or actually as shown here, not just cord blood cells, but also from H1, or from cord blood derived IPS cells, meaning IPS cells that have the VDJ gene and germline configuration, you see here these double positive CD4, CD8 intermediates, as occurs in the thymus. But in the TIPS-derived cultures, we actually don't see these intermediates. At first, we thought they were very evanescent, but very few form. If you wait long, you'll see sometimes 4 or 5% of these forming in delayed fashion. So something is different in the differentiation pathway, and I just remind you here that normally the beta chain is first rearranged, and when a chain is generated, it pairs up with a pre-TCR, which will then direct it to this double positive commitment from which single positive T cells are derived. However, if a gamma delta T cell receptor is formed, then these cells take this pathway. And so we hypothesize that in the case of the TIPS, because the VDJ genes are already recombined, both of them, the cell will behave like a gamma delta T cell, even though, again, it's an alpha beta T cell receptor. So to do that, we knocked out the alpha chain, which leaves, of course, the beta chain capable of pairing with the endogenous pre-TCR alpha, and predicted that, or at least hoped, that that would put these cells back on track to the double positive pathway. Of course, they couldn't make a T cell anymore since they don't have a T cell receptor. We've knocked out the alpha chain. And so this is what you see here. We're comparing the TIPSC and then one of the knockout lines at different days here, 20, 25, 30. So again, the TIPSC induces TCR alpha beta, which you see here on the cell surface at these different time points. And the TIPSC knockout is incapable of doing that because the knockout is effective. However, what we wanted to know is whether these cells would now form double positive cells, and surely they do. So day 20, you see here day 25, day 38. In the knockout, we now see them progressing to the double negative, the double positive stage. So that would not allow us, though, to make a T cell driven by its own T cell receptor. And so we've explored other approaches to achieve this result and focused on the strength of notch signaling. And again, the original line was OP9 using a mouse DLL1 or human DLL1, and we made another one here with expressing human DLL4. And I'll just show you again one slide from this study which shows you that the DLL1 cell line on these TIPS cells is unable to induce these double positive cells, CD8 and CD4, whereas the stronger signal provided through DL4 now again puts these cells on track, and these have their T cell receptor. Furthermore, if you look at these cells now, they induce markers that we never saw before, and one of them is CD1A, for example. They don't express CD56, the marker that you would expect when differentiating these cells towards an NK cell direction. And most importantly and most gratifyingly, we now see for the very first time the induction of CD8 beta. Because the CD8 that was induced, that I showed you in previous slide and that others have reported in the literature, if you check their papers closely, you will note that they always show you CD8 alpha but not CD8 beta. And now, at this time, as these cells now are back on track, we see CD1A and we see other markers such as CD8 alpha beta. So we think we're on the right track now to be able to generate appropriate alpha beta T cells, but what about CARs? Well, I mentioned that the TCR signaling could interfere with lineage commitment, and so can the CAR. Not all of them, but some of them, and here's an example of one that does that. So it's the same TIPS T cell, the same DLL4 that will induce the double positive stage, but now having the CAR there, as you can see, prevents that. Not all CARs do it. Some do, some don't. And I'm sorry that I'm not ready to tell you more about that as of now. And there are many solutions, though, to restore these cells on the proper pathways so as to generate alpha beta-like CAR-expressing T cells. And so clearly a lot of this has to do with the balance between TCR or CAR signals and notch. In the TIPS, with the weaker notch signal and the endogenous alpha beta, the fate was gamma delta. If you took out the TCR alpha beta, you put them back on the alpha beta receptor, and as well as you give them different qualities of notch signals, you can put them back on this pathway. So why do we want to do that? I don't need to beleaguer the case. You've heard about it all morning. We think that this platform will be extremely appropriate to further engineer the cells, and with the goal of precluding their alloreactivity so that they can be administered to more recipients, and of course making them histocompatible such that batches of T cells can be used for multiple donors. So CARs retarget functionally enhance primary T cells. They can induce, certainly in ALL, responses in patients that have failed on all known chemotherapies. They can be engineered to graded levels of potency. 4-1BB ligand is one of several approaches, but that we think results in a far more potent CAR. I didn't go into this today, but they're not just killer cells. Obviously, they can be designed to affect the tumor microenvironment, which we think will be essential for many solid tumors. And we're hopeful that despite the many challenges, that pluripotent stem cells will be harnessed as an alternative source for cancer immunotherapy. I just want to, there are a lot of people to thank in this work. The work in my lab on iPSC is previously performed by Maria Themeli, who's now in the Netherlands, and now taken over by Sjoukje van der Stegen. And my two long-term collaborators on all of this work are Renier Brentians, who now heads our newly established cell therapy service at Sloan Kettering, and Isabelle Rivière, who directs the GMP facility in which T cells, but also ES cells and soon iPS cells are being made at the GMP grade. Thank you very much for your time.